Well, thank you, and I don't know how much you'll hear about glioblastoma, but I think I can possibly spell it now. I would like to thank, in particular, Karen Newell for the invitation to come to this meeting. It's been great, really great stuff, and also for the opportunity to speak to such an audience that tends to be quite different than the ones I usually speak with, which is good. I got enough. Okay, let's see if this works. So I'm pleased to be able to put. Oh, don't tell me this isn't working. Can't even. Let's try this one. All right, we'll try this one. I brought my own and it doesn't work. Anyway, I like to, because I don't get a chance to use this term in our talks, and it's a term created by Karen Newell. When we first started interacting, I had just come to Texas from the University of Nebraska, where I had started a group in comparative biology, we called it, and really it was an excuse for me to spend some time with animal biomedical biologists because I thought it was quite informative and instructive from my point of view, and we don't perhaps cross that bridge enough. But really, in most of my talks that I give, it centers around the idea of death be not proud, or really specifically programmed cell death or apoptosis, which is quite controversial in plants since we're bringing that up. Whether apoptosis occurs in plants is still a bit of a debate, as well as all that's associated with it. So we study those kinds of things in a plant perspective. Okay. Oh, come on. Whoops. So what I'm going to talk about today is our development of this idea of comparative pathobiology with respect to commonalities, if you will, but it's more than commonalities with the way cells do business. And I've argued in a number of review articles that really once you get down to the cellular level, a lot of the players are quite similar, although the context obviously is very different, but it's still very instructive to kind of do these comparative analyses, as I'll try to convince you. Okay. Oops. So plant receptors, and just briefly, this picture makes it a little too good, are quite similar with respect to mammalian and animal receptors, and they have toll-like receptors. They have PRRs, pattern recognition receptors. But for example, just to argue my own point, tyrosine kinase phosphorylation was just reported in plants about a month ago. So knowing what we know about, or knowing what little I know about tyrosine kinase receptors in the mammalian systems, this is a distinct difference. The other point I want to make is that most plant receptors are cytoplasmic. We spend a lot of time looking for more classic receptors. For the most part, these NVS-LRRs, which are a diagnostic motif for plant resistance genes, are cytoplasmic for the most part. However, they feed into a lot of similar types of pathways, as you see here, and I'm sure we're all familiar with many of these in terms of MAP kinase pathways getting activated, ultimately to activate transcription in the nucleus for the appropriate cellular response. This is quite conserved. But what is more unique is what I'm going to try to tell you in the next few slides. And given that this was a cancer-related meeting, I just wanted to mention Agrobacterium, which is responsible for pretty much all of the transgenic plant work that you might ever read about is all done by Agrobacterium. And I just want to say it's a very interesting bacterium because what it does is with the use of, it harbors mammalian promoters and promoter elements very, very distinctly, and uses these elements once it gets into the plant. It's a type 4 secretion system, and it inserts DNA of interest. This DNA of interest in the case of Agrobacterium is novel amino acids. So what the Agrobacterium is doing is hijacking the plant's machinery. These cells are immortalized, they're tumorigenic, they're known as tumor cells, and they are instructed by the bacterium to basically synthesize for the bacterium alone novel amino acids that only this bacteria can utilize. So they have a very nice system. But it's been, again, debated quite a lot of whether you would consider that a cancer, but I'll leave that alone. But it certainly produces immortalized cells in what you can call in galls and/or possibly tumors. And it's a great tool for genetic engineering, which is what everybody, including us, use. Okay, so the story with this topic starts in this nicely infected cornfield in Nebraska. This is stalk rot of corn, and it's caused by a fungus, Fusarium. That is an endophyte, which means it lives inside the corn cell. And under conditions of environmental stress, such as heat, cold, things of that nature. In this case, it's heat and drought. This fungus proliferates, turns pathogenic, and does this. Why am I showing you this? Well, it turns out this fungus produces lots of secondary metabolites, as fungi tend to do. And my background, I should say, is in fungal diseases and plant pathogen interactions. This fungus produces lots of metabolites, one of which is known as fumonisin B1, which is a ceramide synthase inhibitor bound to sphingolipid biosynthesis. But fumonisin B1 is a potent carcinogen. And it is found basically you'll find this fungus in any corn plant that you might have the reason to check out. But it's been now known by the FDA, who did a 2-year study on this, is that all corn-based food products, to varying degrees, have this fungus and have this toxin in them. It's a food safety issue. You go buy a corn tortilla, you will have fumonisin in it. It was discovered in beer in South Africa just 20 years ago, and it's kind of like the new version of the aflatoxin story in the sense that it's a carcinogen. So we were interested in this from the carcinogenic levels and perspective, and early on, this is an old vintage slide. This is a tunnel reaction, very diagnostic program, apoptotic-like cell death. It's not that uncommon in the mammalian systems, but let me just point out that this fungus also infects tomatoes. So we can take tomato cells, we can take monkey kidney cells, treat them with fumonisin B1 and do a time course. These are DAPI stains. This is the tunnel reaction, indicative of DNA fragmentation. It's very well used, again, in biomedical situations. And what you see here with the tunnel stain is basically the formation of apoptotic bodies. If you look closely, which you can't see in this, the membrane is actually, plasma membrane is still intact, very diagnostic. And ultimately, these apoptotic bodies migrate to the periphery of the cell where they're phagocytosed by macrophages. So it's very well, reasonably well understood in mammals. If you take fumonisin B1, do the same experiment in monkey kidney cells, you get basically the same thing. Here you see the apoptotic bodies migrating to the periphery of the cell where they're ultimately engulfed, okay? Now, this alone, I could, and I won't, but I could show you many, many plant reviews from many, many noted plant physiologists concluding that apoptotic bodies do not occur in plants, laddering does not occur in plants, and these diagnostic features in general do not occur in plants. And all I could say is that is not entirely true. So another sort of example as we got into this area is this is alfalfa, and this is an alfalfa pathogen, Colototricum trifoliae. It's not important, but what is interesting is that the gene that's responsible for causing this lesion is the same gene that causes these tumors in mice. And again, at a plant meeting, you don't see this kind of slide, but we had been looking, we had been doing knockouts, and one of the genes we knocked out in the fungus was a rash gene, the first oncogene. And we were interested in understanding, well, how similar is this rash gene, the alfalfa pathogen in Nebraska, how similar is this rash gene to the relatively well-studied rash genes in mammalian systems? And it turns out the hot spots for tumor formation are well conserved. In fact, the protein in general is well conserved. In fact, it's closer to NH3T3 cell rash genes than it is to yeast rash gene. But in any event, they are also functionally active, as you see, with some help from my friends in the biomedical area in Nebraska. They basically taught a very good student of mine how to do these experiments, and essentially we could transfer the Colototricum alfalfa pathogen rash gene with mutations, one of which was a natural mutation that we discovered, and basically transform the cell line and cause tumors in pretty much every mouse that was tried. So the rash gene, the context again is very, very different. Alfalfa plant and a mouse. The fungus clearly has the genetic ability to create oncogenes. Now, so what we did is we did it for the fungus, and we put this oncogene this so-called oncogene right back into the fungus, and studied what would an effect of an endogenous oncogene have on the physiology of this fungus. Clearly has nothing to do with cancer, and clearly is not going to make tumors in this fungus. But it did have a very interesting phenotype, which again, I won't give you too much details because it'll take too much of my time. But it lost polar growth, it lost the ability to differentiate, it was clearly abnormal morphologically. And these phenotypes are only seen on limited nutritional media. When you use rich media, even with expressing RAS extremely highly, there was like no effect on the fungus. It was also non-pathogenic. So it turns out it was a nutritional issue in the fungus, which perhaps again is not that surprising. And in fact, I'll just tell you the punchline. We found out that proline, which is one of our favorite amino acids, actually complements the RAS mutation and bringing it back to wild type. But we've done quite a bit of work on that, trying to understand that better. All right, I don't need to show you this slide at all, other than to say that when you have misregulation of apoptosis, bad things happen. And we've heard about some of these bad things today as well. And again, just to remind everyone, plant audiences, how important the regulation of this process is for obvious health and well-being. And that it's a natural, phenomenon that occurs and is required to occur, as you all know here. Here, this one is more for you guys, just to remind people that programmed cell death occurs in many contexts in plants, sculpting of the hand and things like that, I think you're quite familiar with. But the transpiration stream or xylem formation that occurs where water transpiration moves out of the plant are all a bunch of dead cells forming hollow conduits that are programmed and cued to die on cue to form these developmental structures. Pistil pollen relationships, self versus non-self, as you see here, is actually an apoptotic-like event. This is a root cap sloughing dead cells as it grows. Again, this is an apoptotic-like event. The reviewers like apoptotic-like more than just saying apoptosis. So I'm learning. And the hypersensitive response in plants. Now, this is different from the hypersensitive response that you guys deal with. It's a programmed cell death, a limited programmed cell death that serves to resist the pathogen. Okay. Oh, the last one of these sort of cartoons is shown here. Again, this slide you don't have to say too much about the two major pathways. But what I want to point out is one event, and that is downstream of the intrinsic and extrinsic pathways, is the so-called apoptosome. This has been, again, well studied. It's kind of a recruiting factor for different players in the execution of the cell before the decision is made to die. And APAF1 is one of the key binding elements that binds the apoptosome along with cytochrome C and procaspase 9 to generate a higher order oligomeric structure, which, if cued to go on, processes this caspase 9 to activate caspase 3, and you're dead. Well, it turns out the APAF1 gene family is a quite well-known family of genes in humans. I think there's 8 of them. But it turns out that APAF1 structurally, if you do a blast search, actually is closer to a tobacco resistance gene called N than it is to its siblings in the human genome. So this tobacco genome has an APAF1 homolog, which we fooled around with, which is a resistance gene. It's an NBLRR protein that is very highly similar by that criteria to the human gene counterpart, even though the human gene is part of a rather large gene family. So, and it turns out that a number of plant resistance genes, although it hasn't been played up much, except by us and a few others, basically serves to recruit certain elements and also becomes a higher order oligomeric structure, which ends up making the decision whether to undergo a hypersensitive response, which is a programmed cell death by definition. Okay, so these pathways, as you all know, are highly conserved. This is again mostly for plant types. Just to point out, though, that they're conserved to the extent that you can take a BCL2 mutant, complemented with SED9 and things like that. So they can be, to a degree, very nicely complemented by their counterpart. And basically, you see it's a series of protease activations. This adaptive protein binds what it needs to do to activate caspase 9, or in C. elegans, it's a little more simpler, but SED3 is the homolog of caspase 9, and they all result in a programmed cell death. Now, if you look in plants, which we have done, we have a lot of question marks, there are a lot of black boxes, and it turns out none of these genes have really been identified in plants, which, again, is a problem when you're writing a grant. But at the end of the day, many of the plant processes are in fact programmed like cell death with apoptotic features, including disease, the HR, which I just mentioned, senescence, and a number of abiotic stresses, which is a fancy word for drought and heat and salt, things like that, which with climate change and things of that nature, these are becoming much, much more important, again, for food security and things of that nature. So we went out, of course, to try to fill these boxes, as have others. And certainly the easy way of doing this, which is doing blast-type searches, is not going to work. Now there's lots of genomes in the gene bank, and it's very clear that the easy way of doing it, which we and others have tried using antibodies and PCR and homology searches, have really not yielded much. And in fact, there is a group of enzymes called metacaspases, which are computationally similar to to caspases. And it's very much still under debate from both the mammalian side. There's a number of very heated papers questioning the counterparts' ability to classify it this way. So whether caspases are in plants is also still of debate. But nevertheless, at the end of some programmed cell death pathway, you get the number of regulating features that are a result of programmed cell death and possibly apoptosis, which we now believe is more than possibly. So a number of years ago, we did kind of a really naive experiment, and this is when I was working with fumonisin, which is clearly an animal story, and I'm not really an animal trained biologist. So we had a lot of these genes laying around the lab because we worked out a lot of the fumonisin pathways and targets and things like that. And basically, I had a summer student, and he wanted to learn techniques. So I said, take these genes, throw them into tobacco. You'll do transformation, you'll do blots, and and you'll get an experience. So he did, and as you can see, the genes are from very diverse sources, from humans all the way to insects. But the one commonality among all of these genes is that they're cytoprotective. They're anti-apoptotic genes. They keep cells alive. Bcl-2, as I tell a plant audience, stands for B-cell lymphoma. Clearly an interesting oncogene because it showed another way that cancer can occur. All right, so we put these genes in plants, and we used a fungus for which there is no resistance. It's an economically important vegetable pathogen. It's not important for today. But we made transgenic plants with Bcl-2, and here's the wild type getting demolished, it's tobacco leaves. And here you can see with this plant, which I can tell you is there is no genetic resistance to, we saw this. So I told the student to go back and do it again, probably a half a dozen times. But the important point was he was just trying to, he's a summer student, undergraduate, trying to learn techniques. There was no experimental bias. However, he kept repeating this quite reproducibly over months. We did crosses, we did lots of things, because to come out and say this Bcl-2 would inhibit this particular disease is really difficult to say at the time, especially given what we know what Bcl-2 does. How can human oncogene confer resistance to a vegetable pathogen? We asked that quite a bit. And anyway, after repeating it countless times and doing a lot more work than I'm going to show you, and convince ourselves this pathogen produces oxalic acid, probably kidney stones, but in plants, oxalic acid is a very potent virulence determinant that this fungus produces. It's a toxin-producing organism. It's a necrotroph. It feeds on dead cells. So, maybe what's happening here is you're not allowing cells to die, and this fungus is running out of nutrition. These are agar plugs, running out of nutrition, and without dead cells, basically the ball game is over for the fungus. So we postulated that actually by inhibiting cell death, you're actually inhibiting this fungal disease. And we've shown this with a number of the other anti-apoptotic genes. And again, to convince ourselves, for obvious reasons, giving this talk at certain meetings, got people up. So that just to show that these genes are more, so how do you know, and I've gotten this question before, how do you know that the transfer of this foreign DNA is potentiating a defense system in the plant, and you're just able to catch that? The plant is poised to respond. Well, first of all, if you're a grower in the U.S. growing vegetables, you don't really care what the mechanism is. If you can get your crop in, that's fine. But we were, and we were trying to establish a case not only for the function and expression of this gene, but also for how they might inhibit this particular and other diseases. So we made site-directed mutations in Bcl-XL chicken developmental gene, and we're known to render this gene functional, unable to dimerize, and then made transgenic plants. And here's the control. As you can see, again, it inhibits Sclerotinia, this fungus, very nicely. You can see the white fluff. The fungus is growing out of the agar plug and then stopping, okay? Presumably because it's run out of carbon and it has run out of dead cells. When you make a site-directed mutation, making these null mutants, make transgenic plants, you've lost that resistance. Clearly, the fungus is now able to infect very nicely. So these genes need to be intact. So we were able to conclude a while ago that these brute force type of pathogens, which have lots of cell wall degrading enzymes and things of that nature, actually are a little more sophisticated than that, and they actually hijack and reprogram the host cell death pathways to kill these cells that they end up using for nutrition, which is what they need, what they absolutely need. So we went to a rather naive sort of hypothesis or conclusion that whoever controls programmed cell death, either the pathogen or the plant, wins the battle. And actually, surprisingly, this idea has held up pretty well, somewhat surprisingly. But some simple statement like that has explained a number of different host-pathogen interactions. Okay, come on. So why do we get such, I'm not going to get into this in too much detail at the end of the day here, but why do we get such resistance? Because we also get resistance not only to all pathogenic types of organisms, which I haven't shown you, but also these abiotic stresses, which I'll show you in a second. So we get the same gene to varying degrees can impact both abiotic stress and pathogen attack. How does that happen? Well, there are a number of ways to explain it. In mammalian cells, these Bcl-2 family guys are sort of like cellular sentinels poised to respond to a given stress, where then they can translocate to the mitochondria and do what they need to do to activate defense mechanisms. And this could be what's happening in our case. But one of the things we sort of favor, and we admit all prejudice, is oxidative stress. All of these stresses that you see have been documented by us and others to involve oxidative stress. And in fact, we see oxidative stress as a major intermediate between the challenge or stress and life or death. So we sort of favor oxidative stress, which has also been associated in Bcl-2, and we're spending a lot of time trying to establish that oxidative stress is a major component of the life cell death decision. So we were interested in identifying genes. The next few minutes are going to be a summary of some of that stuff. And we went after abiotic stress because it's a lot easier to control. When you inoculate with a pathogen or a virus, there's a lot of variation. And if we can just use one of these abiotic stresses under a set condition, we can zap tissue or protoplast cells with this particular stress and then look for genes that impact that stress, which will be candidates for anti-cell death types of genes, and we've done that. I'm just going to say anecdotally, this is how we got into sort of abiotic stress in cold. In Nebraska, you might not, most of you from California, many of you from California, it gets very cold in the winter. In fact, it's pretty cold right now. And we had these plants in the greenhouse, which was about a half a mile walk from the lab, and this was over Christmas vacation a number of years ago. And I walked into my daily venture into the lab, and I asked the student what's going on, and said, I blew the experiment. I don't have a car. It's very cold out. I carried these plants by hand for about 20 minutes out there. It was freezing in Nebraska, and now they're all dying. So I said, oh, well, if you looked at the plants a little closer, what you see is all the wild-type plants are dying, but all the Bcl-2 transgenic plants at the time are looking just fine. So realizing it was going to be very hard to replicate that experiment, we were able to confiscate a refrigerator, control the conditions, induce a programmed cell death, and I'm not showing you the DNA ladders and tunnels because we had to do that to publish because the reviewers demanded those things to say programs apoptotic-like cell death. So we've done all that, and I'll show you a couple at the end. But it got us into thinking about abiotic stress because as an economic worldwide problem, these abiotic stresses are actually more, I hate to say this, more important than some of the plant diseases we work on, both economically as well as socially. You're all hearing about global climate change and all this kind of stuff. This is impacting plants heavily, and so there's a lot of interest in defining abiotic stress and development. Monsanto, all the companies are generating plants, we're trying to generate plants, and they're coming out with a cold stress plant in Arabidopsis very soon, creating Arabidopsis. But anyway, so now we were interested in abiotic stress. Oh, come on. So how do we go after genes that impact this? So one is the way I just showed you. Another is just using yeast, and this is nothing new. You have a lot of different yeast constructs. You can take advantage of regulating conditional promoters, and we can express mouse Bax, a pro-apoptotic gene, under galactose promoters. You plate them out on glucose, they're very happy. You induce galactose, which triggers Bax, you're dead. You can take that same construct in same situation and then what we do is pass plant cDNA libraries through this and basically then induce triggering expression with galactose. And again, when you select transformants on glucose, they're very nice. When you induce with galactose, most of the cells are dying. And what you end up getting is it's a nice screen because what you end up getting is just a fairly few survivors, which are candidates for anti-apoptotic genes. We've identified a number of genes this way, then you pass them through a number of different screens. We have different kinds of yeast vectors with part of the pathway recapitulated in mammalian cells, so you can actually also find what spot in the pathway they're inhibiting and things of that nature. So it's an basically easy experiment. but we spent a lot more time in using computational approaches. And what we're looking for is basically structural similarities. Remember, I told you that the DNA sequence level, we were unable to find really anything that was corresponding in plants that corresponded to any animal genes that were of relevance to our interest. So I had a collaboration at the time with John Reed at Burnham, and he had a whole wing of computational people. And every time I go out there, I'd say, you got to do plants, and and the computational guy was not really that interested in plants, but I was a bit of a pain in his neck. And finally, he agreed to do some computational studies with his various threading algorithms, which I won't explain. And what he ended up identifying, or helping us identify, was a corresponding member of the BAG gene family. That just stands for Bcl-2 associated methanogene. They're actually in clinical phase 3 trials as well. They are co-chaperones. So they're not directly anti-apoptotic genes, they bind to HSPs. And they're cytoprotective, it's a cytoprotective gene family, which if you look it up in the literature is actually fairly reasonably extensive, although we hadn't, we weren't aware at the time. So when my student pulled out one of the, from Arabidopsis, the predicted BAG gene, we found a whole gene family with various motifs, and we went on to study this. So, duck? Oh, all right, better good.
Not duck.
Okay. Okay, so we did some modeling, and which is one of the diagnostic features of these BAG genes binding HSP, heat shock proteins, which you see here. We did NMR in solution structure and these banding structures, and this is the one I really want to show you, because without even looking at any of the motifs, you can see from there, I hope, that structurally these guys look very similar. Now, there's less than 10% DNA sequence similarity in this gene, but if you look at the helical structure, water-accessible surfaces, the enzyme sites, basically the structure of the protein is remarkably similar whether you took into the human protein or the Arabidopsis protein here. Okay, and this was good for us because it shows we can make predictions with structural studies that are independent of sequence. And with Arabidopsis, it's very easy to get knockouts and then avail yourself of all the features of this experimental model system, which we did. And again, with time, I'm not going to be able to go through all of it, or any of it. Come on. Okay, so we've localized them. It turns out this BAG clearly localizes throughout the cell. This is just showing what it looks like in the ER, which we've done a lot of studies on. It actually translocates to the nucleus where it activates transcription, this particular BAG gene. And, we have, the BAG, when you knock out this particular BAG gene, it's sensitive to salt. Turns out when you overexpress this BAG gene, it is very, very robust in terms of drought. So this drought experiment is really, really high tech. You stop watering the plant. And what you will do is, if you don't water the plant, it will die. And it'll die in this particular case in a couple of weeks. But when you overexpress this BAG gene, now it's not growing really well, but it is retaining green. And when you rewater it, it's doing just fine. So we've actually had 8 weeks of non-watering, where the wild type is dead in 2, where it actually still is viable. This is important if you're a grower of plants and have weather problems. If you can hold out, I was told by the wheat people, who I know very little about it, you can extend the harvest period 1 week under adverse conditions, you get a 30% increase in yield, which is very, very significant. So but the one that's interesting to us, and I'm going to go through, I have to go through very quickly, is bag 4, which is over there. And what you'll notice in bag 4 is basically it's resistant to this particular fungus, but when you knock this gene out, it becomes very, very susceptible. So we were very interested in what was being knocked out, or how this bag gene was able, when not present, is very, very susceptible. And I'm just going to have to go through it quick because I want to, we're under a time constraint. I'm going the wrong way. So it turns out that this BAG gene, trying to figure out its function was very difficult because it turns out it kills cells. So whenever we did yeast 2 hybrid or things like that, we get nothing, not even back false positives. So we found out that this BAG gene is a lesion mimic. That means it produces cell death in the absence of pathogen in this case. So the brake for PCD has been removed. Okay, that's what we postulate. Interestingly, this gene also has a caspase-1 cleavage site, which is functional. And we think that actually that's involved with processing because of time. I won't show you that, but it does cleave. Come on. This is just showing you that caspase-1, human caspase-1 cleaves this gene. So does the fungus. That's what you see here. And what that means to us is, what it has shown to us is when you get cleavage, you get resistance to the pathogen. The reason by which you get resistance to the pathogen, when this is cleaved by a specific aspartyl protease, you induce autophagy, or the biomedical autophagy. And what we were able to do, that was a good, actually not a bad picture, come on. So this is EEM just to show, here is autophagy occurring in the processed bag gene, and here you see the vesicular structure. It's classic autophagy. And these they have their cargo now. They're actually unloading their cargo. You notice the double membrane diagnostic for autophagosomes, and basically they become ghosts after having delivered their cargo of cellular debris in a programmed type of cell death, in which is induced by the fungus, or is induced by the fungus, where the plant recognizes this, activates this protease, cleaves this bag gene, and you get autophagy and resistance. The wild-type fungus, which is pathogenic, you see none of those markers for autophagy, or those morphological markers. Instead, you see apoptotic type of markers. Here you see nuclear condensation and in the chromosomes, and basically very little disturbance of the cell till it gets to be too late. So the fungus, I'll tell you the punchline, suppresses autophagy, which we were keen on showing, induces in a temporal and kinetic way, induces apoptosis, where it feeds off the dead cells, which I told you with earlier. So suppression of autophagy is also a popular topic in my lab. This is just to show you from the same thing as the EM shot, that the mutant, or this non-pathogenic resistant plant to this mutant induces lysotracher staining, which is not entirely specific. The wild type, you see none. This is indicative of autophagy. This is suppressing autophagy. We've done NDC stain, which is a little more diagnostic and robust. And again, the non-pathogenic fungus induces autophagy and resistance. The wild type, it's clean. It causes disease. Okay. The last part, and we could talk about bananas probably at this time of day. So bananas is a particularly good crop for a molecular biologist. And the reason is if you go to the store and buy a banana, or the plantains we had today, you'll notice, or maybe tomorrow you'll notice, that they don't set seed. There's no genetics for any eaten bananas anymore. So there's really nothing the breeders or anyone else can do. And it turns out when bananas have a problem, like pathogens or environmental stress, people starve. In fact, the starvation and death of many countries where bananas are staple is now well known, and it's becoming a topic of interest because the strains of the fungus that infect this plant are mutating. This was in Science a couple of weeks ago even. They're adapting, and now they're pathogenic to all the cultivars of banana that are commercially eaten. So in countries where your banana is a staple, like Uganda and other places in Africa, the average consumption of banana or banana-based food products, that's plantains, dessert bananas, that's beer, vodka, wine, seriously, everything they consume is banana-based. So when you lose banana production in these countries, which has happened in the past, people do starve, and that's going on right now because of this new race of the fungus, if you will. So there's no way to manipulate the banana genetically, so for molecular type, this business is good, because what you can do is propose to put genes in banana that will confer resistance to these bad pathogens or these bad abiotic stresses. And basically, there's no alternative. Okay, now this slide, oh boy. Okay, so this is a field in Malaysia of Cavendish bananas. You go to the store, you'll see Cavendish. It was put in 20, 30 years ago to overcome another strain of a fungus. Typical of American agriculture, you have a disease problem, you put a resistant plant on, and X years later that is overcome, and it's an arms race. So in this case, the Cavendish was resistant to a former pathogen. This pathogen is now resistant to Cavendish, but a new race of the pathogen. As you see here in Malaysia, this field was totaled in like 3 weeks. It's a very aggressive pathogen, and basically there's no real recourse in terms of control measures, except perhaps transgenic, with all prejudice admitted. So we got some money, actually from the Gates Foundation at the time, to look at this further, and quite simply proposed to put antipatotic genes in banana. So this is banana embryonic cell suspension cultures, and this is how traditionally it's done. You put out a bazillion clones of these guys, and look for the few that may come up positive. It's a numbers game. If you do enough, you'll get banana transformers, but it's very inefficient. However, and this we did look for, when you try to transfer an intact anti-apoptotic gene, which is what we're doing to see for disease resistance to these fungi, we noticed right away that after 4 months, these are slow processes, these transgenic plants, you see basically no black and yellow and green. So the transformation frequency with Agrobacterium is humongous. It's about 50 orders of magnitude, because we quantified this than the controls. Why? We believe it's because of Bcl-X cell, you're suppressing cell death. So let's show you the type of cell death that's occurring. This is DNA ladder. This is good for us. It's very common for you people. But it shows when Agrobacterium is seen by the plant, you get DNA ladder formation and you get, tunnel positives. Classic, and here you see them right here. This is tunnel stain. Here's the merge. So this is banana roots, and as you can see, the plant is responding in an apoptotic-like manner. And that's not surprising at all, because bananas are not only infected by Agrobacterium, it's a non-self-recognition. It recognizes the bad guy being Agrobacterium. It undergoes a cell death regime to delimit this pathogen. Ball game over. So it's a non-host interaction that induces a cell death to protect the plant. When you add an anti-apoptotic gene to keep cells alive, the transformation frequency and plant vigor are correspondingly greatly increased. But the real test is putting seeing if it works against pathogens. Here you see ladyfinger, a common commercial banana with the fungus inoculated. It's a soil disease. It's going down. It's a wilt. Whereas you see, and this is the control. Here are two transgenics. They look just like the control. If you look at the it's a vascular disease. So if you look at the cut open vascular tissue, you can see it's all black. That's indicative of cell death. The fungus is killing the plant, and as a result, water and everything else going upstream is a problem. So the plant is undergoing Panama wilt as the disease. The transgenic plant looks just fine, and these vascular systems are totally clean. This is the fungus. So this is using a worm. SED9 is a C. elegans developmental gene, works just fine. And we have these plants not only in Australia, which is good, I get to go to Australia, but also in Africa and Uganda. We're doing our these are analogous to your clinical trials, these field tests, which generally don't translate that well from the lab, I might add. This is showing drought tolerance, which we also get in these transgenic plants. And again, even though it's a tropical country, they have periods now where they get no water, where the weather system, again, is kind of wacky. All right. So we have those going on in Uganda as we speak. We also have field plots in Africa, in Australia, which is good because it's a nice place to visit. And I'm just going to tell you that the other aspect of all Gates grants for ag crops is raising the carotenoid levels for vitamin A deficiency, particularly in children. And vitamin A deficiency, you may have heard of golden rice, which is the original guys trying to do this, and they've had problems. But vitamin A deficiency in developing countries is a big deal, and there's a lot of fatal problems particularly with children with this disease. So we and mostly my partner in Australia is basically trying to bump up carotenoid levels, and this is doing by metabolic engineering. The idea was to shunt one pathway into the provitamin A pathway. This was not so easy. It sounded good. Took us like 5 or 6 years to work this out. You can't start mucking up pathways in these plants or animals, for that matter, and expect not much to happen. So we had all kinds of problems. Even the source of the transgene, whether it was from banana or from another plant, even though it was the same enzyme, they had very different activities in the transgenic plant. So it was a big pain in the neck, but we got orange bananas, as you see here. These are high carotenoid producing bananas. They're too high. The FDA has stipulated levels which are more like this, but we were trying to see how much we can push expression because that was a big limiting factor, and we were able to obtain orange bananas, but they're way higher. They're in way higher concentrations than necessary. This is more what is necessary. Here you see these transgenic bananas growing looking just perfectly normal in Uganda. Here you see what they look like, and you can, again, by overdoing the expression, you can get these orange bananas, but really all we need is this level of expression, which is perfectly normal. They are now, just about 2 weeks ago, in human trials in the US. Okay, and with that, I'll stop and thank all the people. Oh, we also have it in golf turf. That was more of a joke. I don't golf, but this is a real golf turf. I had a student who was a golfer and wanted to do this. So we actually have transgenic, this is Ben Crenshaw golf. there's nothing terribly we're going to learn scientifically from this, but actually there's, you got to have a green golf course, right? So there's a lot of interest in that, not necessarily by me. And I just want to thank very quickly all the people who helped me. I clearly had a lot of help, and particularly Mady, wherever he is, and Brett Williams, who's left. I guess I took him off, boy.
He's in the next corner.
Oh, he's at QUT now, yeah, but he was in my lab postdoc. Those guys did a lot of the trench work with the bananas that I told you about, and of course everybody else that I collaborate with. Thank you for your attention.